Welcome, everybody. I'm very pleased to introduce our Biochemistry and Biophysics Capstone students who selected fentanyl and the opioid crisis as a socially relevant topic to know more about at the molecular level. About 200,000 scientific papers use the word opioid, so this is a very broad and complex topic. And while we've barely scratched the surface, we have learned a lot and hope you'll come away from this with a basic framework for thinking well about this topic that really involves an intersection of some fascinating biochemistry and neuroscience and our well-being as individuals and a society.
To begin, here's a brief overview of the current U.S. opioid epidemic measured by reported overdose deaths. It began in the mid-1990s with an increase in people taking and dying from prescription painkillers. The second wave, beginning in 2010, was largely due to the illegal drug heroin. Then, in 2013, we see a third wave marked by a rapid increase in deaths from illicitly manufactured fentanyl. We are currently said to be in a fourth wave where illicit opioids, especially fentanyl, are combined with psychostimulants like cocaine to create extremely potent mixtures. Deaths due to opioids continue to rise with around 80,000 overdose deaths in 2021 and 105,000 recorded in 2022, 80% of which involve fentanyl. With that context, our outline for today is: we'll first introduce opioids and their receptors, then we'll discuss their physiological effects, including molecular aspects, and finally, we'll circle back to the opioid crisis and look at how we got here and ways we are moving forward. So first, what are opioids?
So the story of opioids begins with opium. It may be the most ancient form of analgesia, which simply means pain relief, with evidence that in 5000 BC Sumeria, it was known as the joy plant due to its euphoric effects. In 1550 BC, the Ebers Papyrus describes opium as a way to prevent excessive crying of children, as well as having other uses. As opium spread throughout Eurasia, opium got its name from the Greek word opion, meaning poppy juice. Doctors such as Dioscorides included opium in their practice and writings, and the Roman scientist Plinius studied the effects of different dosages and intake methods. Later, Paracelsus made a pill form that he would prescribe as painkillers, calling them stones of immortality. And making a huge impact was Alexander Wood's 1853 invention of the hypodermic needle, which revolutionized the way opioids could be administered. This was also the era in which morphine became available. Frederick Serturner in 1803 was the first to isolate morphine. However, the discovery did not get recognition until a few months later. Suffering from toothache, Serturner ingested what he had isolated and it relieved his pain. Because of its sedative effect, Serturner named the drug morphine after Morpheus, the Greek god of dreams. Over the years, many improved methods of morphine isolation were developed, with recent methods like that from 2015 shown here achieving over 90% recovery. And in 2019, over 400 tons of morphine was produced worldwide. The actual structure of morphine was solved in 1923 by Robert and Goland, and in 1960, the biosynthetic pathway, which is shown here, was deciphered. We recognize here the name of codeine, which is an intermediate in the pathway, but among all these compounds, morphine is by far the most abundant, making up about 10% by weight of raw opium. As these molecules are difficult to synthesize, morphine today is still isolated from opium, and a variety of similar opioids, known as semi-synthetic opioids, are synthesized from either morphine or from thebaine, seen here as a morphine precursor. In 1832, codeine was also isolated from opium. It is now commonly used, but is now primarily made from morphine. Some other semi-synthetic derivatives in use today are heroin, first made in 1875, but only recognized as a more powerful analgesic than morphine in 1898, when it was made in a failed attempt to synthesize codeine from morphine. Additional agonists with analgesic activity include oxycodone and hydromorphone. In 1961, naloxone was made and became an important opioid receptor antagonist or blocker used in research and to reverse overdoses. Buprenorphine, made in 1966, is a partial agonist that, as we'll see later, is very useful for treating opioid addiction and is being increasingly prescribed. Before talking about synthetic opioids, we're going to describe 3 common mouse models used for discovering and characterizing analgesics.
2 landmark papers from the 1940s introduced quantitative, reproducible methods that thousands still use today to test analgesia. A 1941 paper, cited more than 4,500 times, introduced the tail flick assay. As seen here, a light source is focused with the lens, heats the tail, and the longer the mouse takes to move the tail away, the better the analgesia. A couple years later, the hot plate assay was introduced. Here, a glass cylinder is placed on a hot plate and mice are put into the cylinder. Indicators of minor pain are licking and or lifting their front paws, as seen for the mouse on the right, or for more severe pain, jumping to relieve pain from their back paws as well. In 1963, Janssen, who you will soon learn, was the first to synthesize fentanyl. He altered the tail flick assay by placing the tails in a hot water bath and measuring the time it took to move their tails. This was also the first paper to report on the analgesic effects of fentanyl. Such assays provided a way for pharmaceutical companies to screen synthesized compounds to discover new opioids of potential use, many of which look nothing like morphine.
Methadone was an early synthetic opioid made by the company Eli Lilly due to a shortage of morphine during World War II. Levorphanol, made a few years later by Eli Lilly, is more structurally similar to morphine. Finally, the subject of our talk, fentanyl, is a synthetic opioid made by Dr. Paul Janssen of Janssen Pharmaceuticals, pictured here to the left, who was motivated to find an effective alternative to morphine that was powerful and specific, yet safer and less addictive. At the time, fentanyl was the most potent opioid with a potency about 100 times greater than morphine, and it is still widely used today. And fentanyl derivatives, 3 of which are shown here, are now their own class of opioids, each having unique properties and many having potencies as high as 1,000 times that of morphine. Sufentanil and alfentanil are both approved for medical use, but carfentanil is only approved for veterinary use. This brings up the question of how potencies are defined.
As seen here, potencies are determined based on dose response curves, where the x-axis is the dose of the drug and the y-axis is some response, which for opioids is typically analgesia, or its ability to reduce pain. Potencies are defined by comparing the so-called ED50 values, which are the amounts of drug that achieve 50% of a target value or achieve the target value for 50% of the subjects. Measured potencies will vary depending on what assay is used, and from these curves based on a rabbit tooth pulp assay, which we'll describe later, fentanyl is about 150 times more potent than morphine. Given this, you may be wondering what factors underlie the huge range of potency seen.
Let's consider fentanyl versus morphine. In this 2013 study using the mouse hot plate assay, there was a 110-fold difference in their ED50 values for analgesia. An obvious question is if this is because fentanyl binds 100 times more tightly to its receptor, and the answer is no. We'll introduce you to its receptor later, but for now, all you need to know is that it has one. Reported here are a set of binding constants for some opioids, and fentanyl and morphine have almost the same affinity for the receptor. Similarly, looking at oxycodone versus morphine, oxycodone is about twice as potent as morphine, but binds 20 times less tightly. Clearly, other factors must be at play here that are impacting potency. Some possibilities are differences in bioavailability or mechanism of action of the compounds once in the body.
Bioavailability is highly important for fentanyl potency. One informative study compared the ED50 values of fentanyl and morphine when injected into the brain versus when injected intravenously. They used the rabbit tooth pulp assay, which, as seen here, involves exposing the tooth pulp with a 1 millimeter hole. Then, increasing electrical impulses are applied to the exposed pulp until the rabbits lick their teeth, indicating that they're feeling pain. Using this assay, the authors of this study determined that fentanyl was 48-fold more potent than morphine when injected intravenously, but only 3-fold more potent when administered directly to the brain. To confirm that fentanyl is better able to cross the blood-brain barrier, they measured the brain and plasma concentrations after intravenous injections. The results were fairly dramatic, with fentanyl being 23-fold more concentrated in the brain than morphine. This difference is ascribed mainly to a difference in lipophilicity, or oil-like nature of fentanyl.
Fentanyl and its derivatives are indeed known to be highly lipophilic, and their lipophilicities correlate well with their potencies shown here. A common measure of lipophilicity is how a molecule partitions between octanol and water, and as seen here, these lipophilicity values for morphine, methadone, fentanyl, and one of its derivatives, sufentanil, are about 1.4, 120, 860, and 1800. This trend matches well with the trend in potencies, which go from 1 to 10 to 100 to 1,000. And this explains why researchers have a keen focus on making increasingly lipophilic opioids. It's a bit more complicated than that because, as shown here, specific proteins also impact transport across the blood-brain barrier. The red circles indicate the drug, the green ovals indicate the P-glycoprotein that differentially pumps molecules out of the brain, and the orange rectangle represents a putative transport protein that may help fentanyl get into the brain. These proteins may also play a role in why much less morphine makes it across the blood-brain barrier, as seen here compared to fentanyl. Aside from its higher potency and lower cost, there are a few pharmacological properties of fentanyl that make it a more attractive analgesic than morphine in many circumstances.
From the start, it was clear that two advantages of fentanyl were its faster action and its shorter duration. Here is one example from 1970 showing the time course of the analgesic effect of morphine and fentanyl after intravenous injection. The differences are quite clear, and it seems that fentanyl's better ability to cross the blood-brain barrier provides a rationale for its faster action. Another factor contributing to its short duration of action is that fentanyl is readily metabolized through N-dylation to norfentanyl or hydroxylation, and neither of the metabolites has pharmacological activity. And evidence that fentanyl is preferred in practice is usage data. As shown here, it's the dominant medicinally used opioid in Europe and the US, with the US uniquely using a lot of oxycodone. Now, having met the opioid molecules, let's have a look at their receptors.
The 1973 landmark paper that documented opioid receptors in the central nervous system reported on the binding of radioactively labeled naloxone to rat brains and guinea pig intestine. The binding in the brain was very high in some places and near zero in others. In the guinea pig intestine, binding occurred in the myenteric plexus, which is innervated, but not in the longitudinal muscle, which is not, leading them to conclude that opioid receptors are only in nervous tissue. Consistent with this, measurements on yeast, red blood cells, and rat liver all showed no binding. 3 years later came the discovery that there were distinct opioid receptors. These researchers explored the effects of many different opioids of the physiology of what they called the morphine-dependent spinal dog, and found notable qualitative differences between the opioid ketocyclazine and morphine in terms of their ability to suppress withdrawal symptoms, shown here, their impact on body temperature, respiratory rate, and pulse. Due to these differences, they concluded these compounds must target different opioid receptors and named them mu and kappa after morphine and ketocyclazine. Just 1 year later, another group used radio-labeled naloxone and lu and enkephalin to probe receptor binding and ability to inhibit contractions in guinea pig ileum versus the mouse vas deferens. Lu and enkephalin specifically inhibited the vas deferens contractions, and it also bound much more tightly to that tissue, whereas morphine interacted better with the ileum. The conclusion was that the vas deferens had a distinct receptor from that, which bound morphine and named it delta after the D in vas deferens. These three, mu, kappa, and delta, are the well-recognized opioid receptors.
Here we will briefly summarize the main aspects of the human opioid receptors and their different roles. First, noting that each has been linked to pain modulation in some way, as well as psychophysiological reward systems for sex, food, social interactions. They are also all structurally very similar G-protein coupled receptors, which we'll discuss in more detail in just a bit. The main opioid receptor and the focus of our talk is the mu opioid receptor, which causes analgesia, euphoria, respiratory depression, as well as sedation. The delta opioid receptor is most closely related to the mu receptor and is known to regulate neuronal pathways for localized analgesia in the spinal cord, mood, memory, and learning, and positive affect. The kappa receptor, which is not as closely related to the others, regulates general analgesia, diuresis, and causes dysphoria, as well as producing a negative affect. In addition, the fourth closely related receptor was discovered later and is now called the nociceptive opioid receptor. Some prefer not to call it an opioid receptor because despite it being a close cousin of the opioid receptors and also involved in regulation of pain and reward processing, it does not respond to similar agonists. Also, interestingly, its main effect is hyperalgesia or enhancing pain perception. And it is also shown to block morphine signals in an anti-opioid mechanism. So not a lot is currently known about the mechanisms, so we won't be mentioning it further today. In any case, with the discovery of the three main opioid receptors, the race was on to find what the receptors normally bind to. So what are our endogenous opioids?
The discovery of the first of such molecules was reported in two landmark 1975 papers. The two short 5-amino acid long peptides identified were termed enkephalins, a name originating from enkephalos, which is ancient Greek for brain. Starting with the equivalent of 5 pig brains, they isolated less than 1 microgram of the active material. When they determined the structure, they found that they had isolated not 1, but 2 active enkephalins. The two peptides look nothing like morphine and differ by their last amino acid, which can be methionine or leucine, and they are commonly referred to as metenkephalin and leuenkephalin. The assay they had used for opioid activity applied opioids' ability to block electrically stimulated contractions of a mouse vas deferens muscle. As proof of the molecule's activities, they observed anticontractile activity after administering both leu and metenkephalins, as is seen here. They also showed the opioid antagonist naloxone completely blocked these effects. The next year, the now familiar endorphins were discovered, but not until 20 years later in 1997 was an endogenous ligand discovered that was highly specific for the mu-opioid receptor. From previous work, Capstone and co-workers had evidence that 4 residue-long peptides starting with tyrosine proline tryptophan had mu-opioid receptor-specific activity. Very cleverly, to test their idea, they simply synthesized all 20 possibilities for an amino acid at the 4th position. As is shown here, they tested their affinities in black bars and specificities in open bars for the mu-opioid receptor. The result was clear. Phenylalanine in position 4 stood out with about 10-fold higher affinity and specificity, literally off the charts compared to the other peptides. They called this compound endomorphin 1. With their target now known, they were able to isolate it from mammalian brain tissue, and while doing so, discovered another peptide, differing by just 1 amino acid, which also showed high affinity and specificity for the mu-opioid receptor. This was termed endomorphin 2. The endomorphins are at least as potent analgesics as morphine, with pain relief lasting for over 60 minutes. This was exciting as the analgesic effects of enkephalins had only lasted about 10 minutes. Surprisingly, despite the 26 years since this discovery, how endomorphins are made remains unknown, but this perhaps emphasizes the challenges in this area of research. While much more could be said, I'll end this topic with a very simplified summary of the 3 main endogenous mu-opioid receptor ligands. The enkephalins are triggered by pain, inflammation, and stress. They interact best with the delta opioid receptor, but also others, and mitigate pain, regulate the stress response, and intestinal contractions. Beta endorphin is released in response to pain, intense exercise, and so-called pleasure states like sex or eating good food. It mitigates pain and contributes to these pleasant moods like the familiar runner's high. Beta endorphin is about equally specific to the mu and delta opioid receptors, but also interacts with the kappa opioid receptor. Endomorphins are triggered by pain. They help to relieve it and also have poorly characterized roles in the immune response. They're highly specific for the mu-opioid receptor. In addition, as a pretty big surprise, in 2004 and 5, it was shown that we can also make authentic endogenous morphine. It is released in response to infection and may help reduce inflammation. Endogenous morphine targets the mu-opioid receptor. The continuing discoveries related to endogenous opioids and their functions demonstrates just how complex these systems are and how much more we have to learn. Before turning to talk about opioid receptors, we'll introduce you to a useful reference molecule that was modeled after endogenous opioids.
Because endogenous opioid peptides such as the endorphins and enkephalins are rapidly broken down in vivo, experiments to determine the activities of administered endogenous opioids were quite difficult. In searching for a stable delta opioid receptor selective analog, in 1981, a group of researchers made compounds based on leuenkephalin. As shown here, the strategy was to put in D-amino acids and N-methylpeptides in some spots, as these should resist enzyme degradation. The most useful compound they ended up with is this molecule known as Damgo. It is as stable as was hoped, but is not specific for the delta receptor. Instead, it exhibits more than 500-fold selectivity for the mu-opioid receptor over the delta and kappa receptors, and has become a highly used reference molecule in studies of the mu-opioid receptor. Now that we've introduced mu-opioid receptor ligands, let's look at the receptor and how it works.
The mu-opioid receptor is one of the members of the G protein-coupled receptors, or GPCRs for short. We have about 1,000 different types of GPCRs in our bodies, and about 1/3 of all medicines target a GPCR.
So as you can see, they're pretty important. Structurally, all GPCRs have a bundle of 7 alpha helices that is embedded in the cell membrane, with the helices connected by 3 extracellular and 3 intracellular loops. The transmembrane helices, as shown here, are named TM1 through TM7, and there is often a short helix 8 that is inside the cell. Next, you can see that the general function of a GPCR is to transmit a signal through the cell membrane. It does this by binding an agonist molecule from the extracellular side, and that binding event favors conformational changes that allows a G protein to bind to its intracellular surface. Antagonists bind to the same site, but don't trigger the activating conformational change, and so can block the activation by agonists. Let's now look more closely at the GPCR conformational changes upon activation.
Compared here are a ligand-activated receptor in green and an antagonist-bound inactive receptor in gray. Visible in this image is a bound agonist at the orthosteric binding pocket. The activating conformational changes generally include small rotations of TM2 and TM7, shown here, an inward movement of TM5, seen here, and a large outward shift in TM6 as a result of so-called tryptophan toggle residue. This movement opens room for binding to the G protein. Now, here is a cartoon showing that binding event. The activated receptor, as shown here, binds to and activates a specific G protein that has 3 subunits, the alpha, beta, and gamma. In its inactive state, the alpha subunit is bound to GDP. Upon binding to the receptor, the bound GDP is exchanged for GTP, and the beta-gamma subunits fall off. Now, as seen here, the free alpha subunit and the beta-gamma subunit can each interact and activate or inhibit their target partner proteins. The activated partner proteins then cause secondary changes, which propagate the signal. The activated receptor continues activating G proteins until G protein-coupled receptor kinases phosphorylate the GPCR. Then another protein, cleverly called arrestin, binds to the phosphorylated receptor and stops its ability to further activate G proteins in a step called desensitization. The arrestin-bound receptor also is then typically internalized into the cell by endocytosis. Following internalization, the GPCR arrestin complex can either be recycled back to the plasma membrane for further signaling or targeted for degradation in lysosomes. It has also been recently shown for opioid receptors that their signaling can continue from this intracellular position. So now, let's talk about how researchers describe the ability of ligands to activate GPCRs, both for G protein and arrestin binding, which are 2 separate things. Shown here is a plot of ligand or drug concentration on the x-axis versus the response on the y-axis. Full agonists are ligands that create a 100% or higher response. Partial agonists produce weaker maximal activity at saturation. Neutral antagonists or just antagonists bind but do not affect the basal activity and compete with agonists for the same binding site. Inverse agonists actually lower the activity below that of the unliganded basal state. This provides evidence that even the basal state can show activity, and the inverse agonist favors a more inactive conformation. Now, recall that there are both G protein and arrestin signaling pathways. Biased ligands, as shown here, stimulate the 2 pathways to a different extent. With this general background, we will now discuss the specifics of signaling by the mu opioid receptor.
As seen here, the kind of G protein bound by activated mu opioid receptor is the G alpha inhibitory or alpha I protein. Focusing on the alpha I subunit first, we see that it inhibits the enzyme adenylate cyclase from producing cyclic AMP from ATP, as indicated by the blunted arrow. The lower levels of cyclic AMP prevent the activation of protein kinase A or PKA, so it stops it from opening channels that would normally stimulate nerve cell firing. Specifically, it prevents the opening of a calcium influx channel TRPV1, as well as sodium influx channels. On the other side, it turns out that the beta gamma subunits also affect ion channels in a way that further inhibits nerve firing. Specifically, the beta gamma subunits directly promote potassium movement out of the cell and prevent another kind of calcium channel from letting calcium into the cell. The net effect, as shown here, is that the activated mu-opioid receptor leads to more negative charge within the cell and a more positive charge outside. And this charge hinders membrane depolarization, meaning that the nerve is much less likely to fire. Worth noting as a take-home point is that the opioid binding to a nerve cell lowers its activity. So now let's look at a very recent and exciting advance in opioid research.
Just last November, cryo-EM was used to provide the first experimental structures of fentanyl and morphine bound to the mu receptor G protein complex. Bound fentanyl is here in orange and morphine is here in yellow. One notable common feature of their binding is that aspartate 149 forms a salt bridge to each opioid's amine nitrogen, shown here as red dashed lines. This aspartate also interacts with tyrosine 328 and glutamine 126, shown as blue dashed lines. These three residues are in all the opioid receptors and are known as the DQY motif. Also, two key differences are revealed. First, fentanyl's phenylethyl moiety, circled here, extends into a pocket between transmembrane helices 2 and 3 and forms interactions completely absent in the morphine-bound structure. And second, at the other end of the molecule, fentanyl's aniline ring also extends further down into the receptor core region, where it makes a direct interaction with tryptophan 295, indicated with a magenta dashed line, while morphine is a bit too far away to form direct interactions. Recall that tryptophan 295 acts as a toggle switch residue to induce the conformational change that activates the receptor. These additional interactions may contribute to fentanyl's higher potency. In 2023, the same group solved a structure of the mu receptor G protein complex with its natural ligand, endomorphin. Here's an overlay of endomorphin with morphine, and below it is fentanyl with endomorphin. The endomorphin structure conserves the DQY motif, and the endomorphin interaction with aspartate 149 is nearly identical with that made by morphine. And seen here in the fentanyl overlay is that the phenylalanine residue on endomorphin, which is known to be crucial for binding, overlaps well with fentanyl's phenyl ring. So, endomorphin's binding mode has similarities with both fentanyl and morphine. With the experimental structure in hand, one interesting thing we did ask was about the accuracy of a couple predictions of the fentanyl binding mode that were published in 2021.
Shown here in purple and green is the experimental structure of fentanyl bound to the receptor. Recall that an aromatic ring here occupies a minor pocket and that the aniline ring points downwards towards the toggle switch tryptophan. This first prediction is shown from a similar point of view, and the prediction is pretty good. The minor pocket occupancy is correct, and the other end of fentanyl is in the right area, but the detail is wrong in terms of how it approaches the tryptophan switch residue. In the second prediction, which was published in a higher profile journal, there is actually not much correct. Although the view they published is different, it is easy to see that the fentanyl molecule is basically upside down. Thus, a little take-home message here is that while the molecular dynamics simulations can be valuable and pretty accurate, they may also be inaccurate or unrealistic. An additional point of consideration was to ask if the structure gives insight as to what leads a naloxone to bind as an antagonist. As an antagonist, naloxone must bind with high affinity, but not trigger the receptor conformational change. It is known that the key feature that makes naloxone an antagonist is its additional allyl group found on nitrogen, an N-allyl group, which is the origin of the prefix nal. Shown here is how naloxone is derived from oxymorphone in just the same way that the antagonist nalorphin is derived from morphine. From the structure, it is easy to suggest that the additional N-allyl group could stop the molecule from binding deeply enough in the pocket to cause activation. A recent modeling study predicted how morphine, shown in orange, can penetrate deeper than naloxone, shown here in blue, and that the residue isoleucine 322 was involved in the difference. To test this idea experimentally, they made an isoleucine to alanine mutation and showed that with this change, naloxone went from being an antagonist, shown in black, to an agonist that activates the receptor just like morphine does. These results increase our understanding and can help guide the design of even better antagonists. Now one might ask, is there evidence that naloxone does not induce conformational change? And as of this year, there is. In March of this year, a preprint compared the measurements of the mu opioid receptor conformational changes that take place when naloxone and a fentanyl derivative bind. Recall that TM6 moves a lot during activation, and so they measured the TM4 to TM6 interhelical distance shown here. Naloxone binding did not change the distance at all compared with the unbound receptor, while low fentanyl greatly increased the interhelical distance, corresponding to hyperactivation. Thus, naloxone can be thought of as a high affinity anchor that holds the mu opioid receptor in its resting and active conformation.
To recap this section, opium has long been a natural remedy for pain, driving the interest to isolate the chemical now known as morphine and to make semi-synthetic and synthetic compounds with related activities. Many of these had substantially higher potencies as analgesics, and some blocked analgesia, including what would become the life-saving antagonist naloxone. Opioid receptors were found to be inhibitory GPCRs highly concentrated in nervous tissue, and learning about these receptors led to discovery of endogenous opioids. Next, we will turn to consider the physiological effects of opioids.
To understand how opioids inhibit pain, we must look at how we receive and interpret harmful or noxious stimuli. The nerves that detect noxious stimuli are called nociceptors, pictured here as free nerve endings in our skin. One of the first ion channels discovered that converts noxious stimuli to electrical current, which nerves use to communicate, was one that responds to the irritant in hot peppers called capsaicin, a discovery recognized by a 2021 Nobel Prize. The red colors in this panel show that this receptor, called TRPV1, is indeed present in the nociceptive neurons. And this panel shows how TRPV1 responds at 50 degrees Celsius, a temperature we experience as painful, but not at 40 degrees Celsius. Since this discovery, several more TRPs have been found. TRPM8 responds to noxious cold, TRPA1 to chemical irritants, and mechanoreceptors to noxious pressure. Activation of these receptors results in a flow of ions into the nerve that triggers it to fire. This signal then propagates down the nerve to the dorsal horn of the spinal cord. As seen here, different nerve fibers synapse at different distinct layers of the dorsal horn called laminae. For instance, A fibers terminate in laminae 1, 3, 4, and 5 and respond to fast pricking pain. C fibers terminate in laminae 1 and 2 and respond to dull, unlocalized pain. From the dorsal horn, secondary nerves carry the signal to the brain. These pathways were discovered using a cool technique called retrograde signaling, where a tracer is injected into a part of the brain and traced back to establish lines of neural communication. One pathway, shown here in blue, goes to the amygdala and then to the insular and cingulate cortices. This is responsible for our emotional responses to pain. Another track, shown here in green, goes to the thalamus and then to the somatosensory cortex. This pathway signals where the pain is coming from and how intense it is. Lastly, the ascending pathway also targets two regions of the brainstem, the periaqueductal gray, or PAG, and the rostral ventral medulla, RVM, shown here in yellow. These regions can send signals back down to the spinal cord, a key mechanism for pain modulation by both endogenous and medicinal opioids.
To give a simple version of how opioids produce analgesia, we show here a blow-up of the dorsal horn of the spine that you've already seen, with the incoming nerve from the nociceptors and the outgoing nerve to the brain. Not mentioned before is that there's also inhibitory nerves shown here that descend from the brainstem, namely the PAG and RVM regions. Due to inhibitory GABAergic interneurons in the PAG and RVM, the descending nerves are normally inactive, allowing pain signals to be transmitted. However, when the opioids bind, the nociceptive signal is inhibited in three ways. First, the GABAergic interneurons become hyperpolarized, thus inhibited, which disinhibits descending nerves from the PAG and RVM, and they release inhibitory neurotransmitters that cause postsynaptic inhibition. On top of this, Both neurons in the dorsal horn also have opioid receptors, so an opioid agonist inhibits both presynaptic neurotransmitter release and postsynaptic firing. This triple form of inhibition greatly decreases the relay of nociceptive signals to the brain, hence blocking our ability to sense pain. Generally, this works very well, as with high enough opioid doses, the analgesic effect is strong enough to conduct surgeries and many other things that are part of miracles of modern medicine. Now that we know more about how analgesia works, let's look at some of the side effects that cause problems.
Unfortunately, in terms of its use as an analgesic, opioids do cause quite a bit of side effects, such as those listed here. While some of these side effects may be desirable in other circumstances, such as causing constipation, which can be useful in treating dysentery, today we'll focus today on some of the more adverse side effects, which are highlighted in bold, that especially contribute to opioid crisis and the loss of life. These are the dependence, tolerance, and addiction noted here, and also respiratory depression and muscle rigidity, the latter being worse for fentanyl than other opioids. We'll now begin with the euphoria, tolerance, and dependence that contribute to addiction.
Let us start with some definitions. Tolerance is change that lead to needing more drug to achieve the same response, while dependence is the need to keep taking drug to avoid withdrawal symptoms. And addiction involves drug craving and compulsive use. It's much more complex than the other two. For a highly simplified look at what leads to these things, we'll start with two regions of the brain. The ventral tegmental area, or VTA, is involved in reward cognition and releases dopamine to the nucleus accumbens. Here is our pleasure center. Normally, GABAergic nerves in the VTA inhibit the release of dopamine, and opioid binding, as shown here, block the GABA release. This inhibiting the dopamine neurons and flooding the nucleus accumbens with dopamine. This both induces an intense euphoria and the formation of strong memories associated with whatever provided that feeling. To understand dependence and withdrawal, we need to also consider the locus coeruleus, a part of the brain that controls arousal and vigilance by releasing noradrenaline depending on its level of cyclic AMP. With this introduction, we can take a simplified look at some of the biochemical change associated with tolerance, dependence, and withdrawal. Recall that opioid binding to its receptor leads to an inhibition of the enzyme adenylate cyclase and a decrease in cyclic AMP. Shown here for a nerve cell is in red, the resting levels of adenylate cyclase, and in blue, the resting levels of cyclic AMP. Normally, the locus coeruleus makes enough cyclic AMP to release enough noradrenaline for normal alertness and respiration. As shown here, opioid use inhibits adenylate cyclase and sharply decreases the amount of cyclic AMP. So less noradrenaline is released, leading to sedation and respiratory depression. With continuing opioid use, the neuron adapts by making more adenylate cyclase to make enough cyclic AMP to offset the impact of drug and release roughly normal amounts of noradrenaline. The additional adenylate cyclase decreases the intensity of the opioid effects, explaining tolerance. This also sets up to explain withdrawal. As seen here, when opioid use stops or naloxone is given, the extra adenylate cyclase will make abnormally high levels of cyclic AMP, leading to excessive release of noradrenaline and withdrawal symptoms such as jitters, muscle cramps, and anxiety. Now let's look more closely at side effects of respiratory depression mentioned above, that is a major factor in opioid lethality.
As shown here in a 2019 study, a typical dose of injected fentanyl, heroin, or morphine can lead to a 50% decline in the volume of air breathed within about 1 minute for fentanyl and about 10 minutes for morphine. Such a drop in breathing is enough to cause death. Adding to the danger of fentanyl is its higher resistance to being reversed by emergency treatment with naloxone. This figure shows that a standard naloxone dose was enough to rapidly reverse the respiratory depression caused by morphine, but not by that of fentanyl. Instead, a 10 times higher dose of naloxone was required to rescue the fentanyl-induced respiratory depression. This contributes to the greater danger of fentanyl. And beyond this, the danger of fentanyl is further compounded by its ability to cause a condition known as wooden chest syndrome. In the early days of fentanyl anesthesia, a case study showed that 499 of 500 patients given fentanyl developed muscle rigidity, and that faster injection and higher doses strengthened these effects. This included a phenomenon known as wooden chest syndrome, involving rigidity of the diaphragm and spasm of the larynx. This side effect is well known and dealt with in hospitals. To quote a 2019 review, anesthesiologists have been safely administering fentanyl to surgical patients for over 50 years and routinely treat the symptoms of rigidity and airway compromise with muscle paralytics and endotracheal intubation. In a later study, out of 12 healthy volunteers given fentanyl, half of them developed muscle rigidity within 10 minutes. This also shows that even when given the same dose, different people experience different effects. From what has been observed in fentanyl-induced deaths, researchers conclude that its unique ability to cause wooden chest syndrome, along with the more typical respiratory depression, makes it especially deadly. One observation is that witnesses of fentanyl overdose deaths sometimes describe signs of wooden chest syndrome, such as stiffening of the body and gurgling sounds. Another is there are often very low levels of fentanyl metabolites in the blood, indicating that death follows just seconds or minutes after injection. Specifically, one 2016 report found that no appreciable concentrations of norfentanyl could be found in 20 of 48 overdoses. Another indication that death can ensue rapidly is that victims often still hold syringes after death. And finally, fairly direct evidence that wooden chest syndrome is sometimes present is that EMTs often report finding CPR more difficult to perform because of their rigid chest muscles. As mentioned in the papers we cite, most users are unaware that wooden chest syndrome exists and how it makes fentanyl especially deadly. Shifting back to molecular mechanisms, it seems that the mechanism by which fentanyl leads to wooden chest syndrome is still unclear, and also there's a lack of clarity of the mechanisms underlying the more general opioid side effects.
So having gone through this severity of these side effects, I want to draw attention to a figure from a 2022 paper that presents the state of the field. It shows that G protein signaling is responsible for analgesia, and that the jury is still out about the relative contributions of G protein and beta-arrestin to cause side effects such as respiratory depression, drug tolerance, and constipation. It seems some older reports implicate beta-arrestin as a cause of deleterious side effects, while more recently there have been studies that call this into question. Let's take a look at some key evidence to get a better idea of both sides of the story. One 1999 experiment involves both hetero and homozygous mice with the beta-arrestin gene knocked out. Using a hot plate assay for analgesia in response to morphine, both knockout mice showed higher and more prolonged analgesia, indicating beta-arrestin is not important for causing analgesia, but is responsible for a desensitization that somewhat decreases and shortens the length of the analgesia. So what about side effects? In a follow-up study performed by the same group, the beta-arrestin knockout mice showed a remarkable absence of buildup of tolerance to chronic treatment with morphine. As seen here, wild-type mice experienced less analgesia on each successive day of treatment, while the knockout mice did not. A subsequent study showed that the beta-arrestin knockout mice had a much lower rates of respiratory suppression compared to wild-type. It's easy to see how these observations would lead one to think that making an opioid that triggered less beta-arrestin interaction would help alleviate side effects. So what recent studies have called these conclusions into question? In this 2019 study, instead of a beta-arrestin knockout mouse, they made 3 versions of mice in which various potentially phosphorylated residues in the intracellular part of the mu opioid receptor were mutated to alanine so that they could not be phosphorylated. The mice were called S375A, 10STA, and 11STA, as this should be another way to abolish beta-arrestin recruitment and achieve G protein biased signaling. When these mice were given either fentanyl or morphine, as seen here, both respiratory suppression, shown here, and constipation, shown here, occurred at levels equivalent to that of the wild-type mice. These apparent contradictory results have not yet been resolved, leading to uncertainty in the field. Despite this uncertainty, researchers are still looking for developing new opioids with better properties by modulating beta-arrestin recruitment.
The recent structures determined for fentanyl, here in magenta, bound to the mu opioid receptor, led to a proposal that beta-arrestin is recruited through interactions with just 3 amino acids, here in orange. Mutation to any one of these to a smaller amino acid absolutely abolishes beta-arrestin recruitment, even at high fentanyl concentrations. This information has allowed researchers to design new fentanyl derivatives so alternative pathways would not be activated. The idea was that a shortened carbon chain of the derivative, circled in red, compared to the fentanyl alanine ring, would not interact with tryptophan 295 or the other 2 amino acids. In short, no amino acid interactions means no arrestin binding. Computer simulations of the design derivatives revealed that the shorter carbon chains do not interact with tryptophan 295. In camp activation assays, both fentanyl derivative 1 and FBD3 matched or had up to 50% of fentanyl's effects, implying that they will have the desired analgesic effects. In contrast to fentanyl, both derivatives show little or no beta-arrestin recruitment. In the author's words, together, our results revealed molecular details of the structural factors important for arrestin activity of mu opioid receptors and provide structural templates for the design of potent and potentially safer analgesia for pain treatment.
To recap this section, nociceptors receive noxious stimuli and communicate this to the central nervous system. And opioids inhibit this communication in multiple ways to produce analgesia. Opioid action in the ventral tegmental area triggers dopamine release in the nucleus accumbens, leading to euphoria and providing a basis for addiction. Opioid action also causes biochemical changes that underlie tolerance, dependence, and withdrawal. Among other side effects, respiratory depression is dangerous for all opioids, and fentanyl seems to uniquely cause a muscle rigidity that makes it even more lethal. And finally, while there is controversy about what signaling pathways lead to what effects, new structural information has invigorated a search for biased agonists that have fewer unwanted side effects. Now that we've learned more about the molecules and processes underlying the opioid crisis, let's circle back and consider some major factors contributing to the crisis and what's being done now to combat it.
Circling back to the beginning, a key factor fueling the opioid epidemic was the pushing of opioid prescriptions by pharmaceutical companies. This overprescription began around 1996 and is closely tied to the addition of pain as a fifth vital sign. Vital signs are measures of the body's basic functioning and are commonly used in the detection and monitoring of medical problems. When declaring pain as a fifth vital sign, the president of the American Pain Society explained, quote, "Vital signs are taken seriously. If pain were assessed with the same zeal as other vital signs are, it would have a much better chance of being treated properly," end quote. A review article in 2018 by Shear and colleagues found that this classification played a role in leading hospitals to prioritize pain management, thus driving the opioid epidemic. According to another review article by Marks in 2020, at the same time in the mid-1990s, major pharmaceutical players were using aggressive marketing strategies to encourage physicians to prescribe opioids beyond their approved use. In recent years, a handful of major pharmaceutical companies have paid settlements for findings that they downplayed the dangerous effects of their products. In one case, the company Insys created a fentanyl-like drug spray known as Subsys, approved for use only in terminal cancer patients. The court ruled that Insys was found guilty of overseeing a scheme to bribe physicians to prescribe sepsis for non-approved reasons, like chronic pain management. Such efforts are understood to have driven the increase in deaths from prescription opioids, shown in the left graph in blue. Now, let's shift to consider what factors are involved in the increase in deaths due to fentanyl starting in 2013. This dramatic third wave spike correlates with a huge increase in the supply of illicitly manufactured fentanyl coming from China and Mexico. The graph on the right shows a 1,000-fold increase in the amount of fentanyl seized at the U.S. border, a measure of fentanyl entering the U.S., between 2013 and 2018. Seizures have only continued to skyrocket, with around 12,700 seizures in 2022. A 2019 review by Mars and colleagues notes that the factors attracting international suppliers to the illicit fentanyl market were the increasing demand in the U.S. for opioids, minimal criminal penalties for manufacturing fentanyl in some countries, and fentanyl being much cheaper to produce than heroin. Furthermore, the high potency of fentanyl means less is needed for each dose, making it easier to smuggle. Like I mentioned, this wave was also driven in part by a high demand for opioids. When describing the demand for opioids, that same review by Mars and colleagues observed, quote, "Some users seek out fentanyl despite the risks, and others try hard to avoid it," end quote. In other words, some users were seeking out fentanyl for its higher potency, but many thought they were taking heroin or standard oxycodone pills, not knowing the actual contents of the drugs they were using. Indeed, the CDC has reported that of the fentanyl-laced counterfeit prescription pills they analyzed, in 2021, 4 out of 10 contained a lethal dose of fentanyl, and in 2022, it was 6 out of 10 pills. So, what are some paths forward?
Solving the opioid crisis will be difficult and complex, involving healthcare, housing equity, assessing unmet treatments and service needs, and much more. Increasingly, approaches to address the crisis focus on what is called harm reduction, aiming to minimize the negative consequences associated with drug abuse rather than seeing abstinence as the only solution. Some of the first harm reduction programs began in the late 1980s during the acquired immunodeficiency syndrome, or AIDS epidemic, and were helpful for both treating drug abuse and reducing disease. But harm reduction approaches have been slow to be accepted in the US because, as noted in this quote, even though drug researchers generally viewed substance use disorders as a disease, in contrast, the general public tended to view substance use disorder as a moral failing. Some harm reduction strategies are needle exchange programs, providing clean, sterile needles, and supervised injection programs, providing a safe place to inject illicit drugs where medical professionals are present. According to the CDC, users of syringe service programs are 5 times more likely to enter drug treatment programs than those who didn't use the syringe service. Syringe programs also decrease diseases and infections such as HIV and hepatitis and limit discarded needles in the streets as they are properly disposed of. Another resource is Naloxone, which was made available over the counter on March 7th of this year, allowing individuals to be prepared with an emergency drug overdose treatment. Another resource of harm reduction is fentanyl test strips, which is especially useful because, like we mentioned, many people are unaware of the true content of the pills they are taking. One additional harm reduction strategy is to recognize opioid use disorder as a medical disorder rather than a criminal problem and provide effective long-term treatment.
In terms of options, buprenorphine is an underutilized yet effective treatment for opioid use disorder. In December of 2022, President Biden signed a bill to eliminate the mandate that health providers need special waivers to prescribe buprenorphine. This waiver had also enforced a cap on the number of patients that could be given prescriptions. As noted here by Lindsay Vuolo from the Partnership to End Addiction, the waiver requirements serve to limit access to an effective treatment for opioid use disorder during an unprecedented opioid crisis. Now we'd like to show some evidence supporting buprenorphine's effectiveness in treating opioid use disorder. Here, researchers were testing which doses of buprenorphine were most effective at reducing opioid cravings over a 5-day period. The blue curve is for the lowest dose at 32 milligrams, orange is 64 milligrams, and gray is 96 milligrams. All doses were remarkably effective at reducing cravings, but higher doses worked faster, achieving craving scores of less than 1 by 2 and 3 days of treatment rather than 5. In this next study, researchers asked how well buprenorphine reduced withdrawal symptoms. As you can see, individuals receiving buprenorphine had far fewer withdrawal symptoms than those who did not. Finally, this last figure shows results of a year-long randomized trial in Sweden asking how well buprenorphine helps keep people in treatment and prevent relapse. This line with boxes represents the 20 control patients who did not receive buprenorphine. By about day 60, all 20 of them either dropped out voluntarily or were no longer allowed to participate due to a relapse of opioid use. In dramatic contrast, of those receiving buprenorphine daily, just 1 voluntarily chose to withdraw and 4 were no longer allowed to participate due to a relapse, leaving 15 patients, a remarkable 3/4 of the cohort, continuing treatment and continuously testing negative for opioids for the entire year. This supports the effectiveness of buprenorphine as a long-term maintenance treatment for patients with opioid use disorder.
In December 2020, Cal Epstein of Beaverton, Oregon, bought OxyContin pills off an online drug dealer. 5 days later, Cal was dead. The counterfeit OxyContin pills actually contained a lethal dose of fentanyl. We've seen in this presentation, and perhaps in our own lives, how prevalent fentanyl has become in overdose cases, and Cal's death and his parents' advocacy have sparked a surge of fentanyl education initiatives in Oregon. Following this local tragedy, Beaverton School District launched a counterfeit drug awareness campaign called Fake and Fatal that offers expert perspectives on the dangers of fentanyl through community conversations and through free downloadable health lessons that schools can implement into their curriculum. Last December, the Oregon Health Authority and the Oregon Department of Education co-developed a fentanyl and opioid response toolkit for public schools. This focuses not only on education about the prevalence and dangers of opioids, as well as the signs of an overdose and the treatments, but also on having schools be prepared with emergency naloxone to save lives. In an interview with Oregon Live, Cal's parents, John and Jennifer Epstein, urge Oregon school systems to honor their son through education and awareness about the dangers of fentanyl.
To summarize, as evidenced throughout the talk, fentanyl is a dangerous molecule that can kill. The propensity for fentanyl to cause chest muscle rigidity, along with the heightened potential of overdose, make it a clear double threat. The history of the opioid crisis makes sense given the overprescribing of opioids as a response to pain assessment, plus misleading and aggressive marketing in the late 1990s. While much was discussed, this talk was merely a brief foray into the extreme complexity and adaptability of our biochemical systems. All natural and synthesized opioids until now exhibit unwanted side effects. But the recent cutting-edge structure determinations of the opioid receptors with bound morphine and fentanyl, as you've seen, can powerfully inform strategies for synthesizing better opioids. The availability of naloxone as an antagonist that blocks opioid binding to the mu receptor is a crucial safety net in a time of national emergency, and it's a powerful tool in combating the dangerous side effects of fentanyl. There are many paths forward to curtail this epidemic, with education and prioritizing harm reduction at the forefront. We hope that today you've gained insight into this pressing issue, even as we have, and that you'll leave here knowing that there is hope for the future.
I'm really proud of these students who have dug in and experienced firsthand how deep and complex and sometimes internally contradictory the scientific literature is, but also how much can be learned from it. We're grateful to the many researchers who did the work we've gleaned from and who are helping provide society with strategies and tools to combat the opioid crisis and increase people's wellness. We also thank the BB department for the environment and training it's provided, and thank you all for your attention. And now we're glad to take questions.